All right, so let's take a deep breath. That concludes the analysis of this particular paper portion of the video. We didn't analyze every bit. We've left some for class, and we've also left some for you to think about in your written work for the homework component, and that's what we're going to talk about now. Our main hope is that at this point you feel much more confident with the article and have learned some ways to parse through a scientific manuscript to make it feel a little more accessible and not as scary, which it may have when you first picked up this article. I remember feeling that way, and I still do. Reading science is challenging, and so learning the formula for how to think about it and how to make your own determination of what to think about these results is so important. It's really to me, one of the most important things you could do with your bachelor's degree is learn how to be a critical thinker in all contexts, and of course this being one of them. And so you can kind of ask yourself, did you enjoy that process? Did you enjoy sleuthing for clues through an article, seeing how the scientific method was employed, how all of the elements of experimental design came to be? Because if so, you should approach one of your instructors and we can discuss with you some opportunities that you might be able to take part in during your undergraduate, or even point you towards some postgraduate opportunities, whether those be graduate programs or jobs, so that you can use these skills and continue to learn more about them to see if this is something you might want to do for your career. And having said that, I want to give you some overview on this week's writing assignments for both the homework and the discussion we're going to have in class. And this is kind of then going towards now putting it back together. We spent some time looking at all the puzzle pieces up until this point. And now we want to put those puzzle pieces together. We're going to synthesize all this together by summation. And we're going to do this in 3 different ways. You've already now in class been tasked with writing the abstract for the article. And that was a challenging thing, no doubt. So that was one form. And so we're going to build on that. But we're going to ask you to do these 3 things. The first is to create a short, abbreviated, written synopsis, let's say, of the whole paper, and we want you to do so for a scientific audience. The goal here is to, in just a couple of short sentences, in your own words, be able to convey the main findings and the main significance. So the main findings and the main significance for this article, as if you were writing to fellow scientists. Learning how to condense an entire story into a couple of short sentences is a very, very important skill that all of us scientists must learn. And it applies to many situations that you might not be thinking about. Like, for example, someone might ask you at some point, well, what do you study when you're doing undergraduate research? And you don't have a lot of time and you don't want to go on and on. So learning how to briefly describe sort of like an elevator pitch version of your work is key for a lot of different places. And so we're going to do this in the first part, and there's more directions for this on the Canvas platform. The second component is taking the scientific audience synthesis you did, conveying those main findings and biological significance of the story again, but in this instance for a lay audience. And this is really challenging. This might sound like it's the easy part of the assignment, but actually this is really hard. And so we're going to give you a link online to a database of the most commonly used words. I think it's the 1,000 most commonly used words in the English language. And you're going to copy paste your writing in here and check to see if you have a version that fits within these words. So we're going to have a very strict word category so that you are learning how to discuss the findings of the paper with someone who might not be a scientist, might not be a biologist. I think this is one of the most important jobs of a biologist, and that being to share your knowledge with people who might not be as lucky as you to be earning a bachelor's degree in this field, and so that they understand the world around them as much as you do at such an advanced level. This requires us to change the way we speak a little bit. We've spent so much time in this class learning the scientific language for reading and writing, and that's great. That's going to be your job and your career. But it's a very different thing then, of course, from how we would communicate this maybe with friends or family who just aren't in this field of study. And look how hard it was to learn all this. So people who haven't then are relying on you to provide them this information. And just as a quick little related note, if we don't, as biologists and scientists more broadly, talk to the public about what we're doing and why it's important, then we're going to continue to struggle with a lot of things like antibiotic resistance and people understanding and appreciating why and how we can tackle this problem. The majority of scientific funding to put instruments and people in labs and do research is funded by taxpayers, the general public. And so we have to convince them that this is important, show them why they should care, and help them realize what we're doing so that they do have an appreciation at that level. So we're going to have you write a lay audience summary, and that actually is going to be harder than you think. And I actually very, very much encourage you this week when you're doing this is to write the scientific summary and write the lay audience summary, and then read them to your roommates, your family, your friends, your BFs, your GFs, all the people around you, and get some feedback and see what they say. See how much they understand of the scientific one, and then if they totally get it when you flip over to the lay one, that would be fantastic. The third way we're going to have you synthesize is by making what's called a graphical abstract, and I'm going to explain that in the next slide. And this is actually becoming a more and more commonly used format for journals. They're going to publish a scientific manuscript, and alongside it is going to be a graphical summary of the article. So this is to summarize the article in a visual way. And basically what you can think about is taking your abstract that you wrote and putting it down, and then side by side bringing it to life through graphics, and we're going to give you some options for how to do this. You can hand draw it in color, you could use a Adobe Illustrator program, you could use Microsoft PowerPoint. Examples are coming here in just the next slide, but this is a really, really effective way. I'm a visual learner. I usually don't understand anything until I see it drawn, or especially when we're talking about mechanisms, having this lead to this with an arrow, and then how does this get to the big picture. That's what we're going to have you guys do, each of these three things. So I'm going to go in the next slide here about more information for graphical abstracts. Okay, so behold an example, and again I've given you the reference down at the bottom with the DOI value, of a graphical abstract as a means of scientific synthesis and communication. And one of the reasons we want to have you do all of these different components of the scientific process throughout this class, including this one, is so that you begin to realize strengths you may have and weaknesses. And we want you to go follow up on your strengths and as indicators of potential career paths that any of these individual skill sets involved in science might be exciting for you to try. And then for the weaknesses, to help you identify areas of need of improvement, and potentially ways of you filtering out things that you don't enjoy doing as ways of eliminating potential career paths. Science is not a one-size-fits-all career. It's not all lab bench. It's not all healthcare. There are so many things you could do. This actually plays right into something called science illustration. And I love this because many journals now, and we learned about peer review and journal publication in the last video, many of these journals are now requiring scientists to publish a graphical representation of their story, a graphical synthesis. And some journals even have staff now to help with this. And you've seen some good graphical representations in textbooks and through Google image searching of a concept. In fact, as a little side tip, the journal Nature Reviews, which is a review article, not a primary literature article, but those review articles come with several beautiful graphical synthesis figures of different mechanisms and concepts. And so as a student, one of the little study tips I recommend is when you get stuck on a concept, go to Google, enter that concept, and then type in the word Nature Reviews after it. So concept and nature and reviews, and then do an image search and see if you can get a really nice synthesis summary figure to help your studying. Because I was a visual learner and I actually really enjoyed visually presenting data, and you might be too. And you might not have even known about these tools. Here is one such version. This actually came in a complex, super challenging article that was very mechanistic in nature from the journal Cell, Host, and Microbe. And this was published earlier this year. And it shows you sort of the whole mechanism. This is actually a story about the skin and the bacteria you're going to find on the skin, and how vitamin A has a big role in sort of how the resident microbiota, not of the gut, but of the skin, sort of takes shape, and then how it may or may not lead to pathogenic infections, in this case of streptococcus. It was a really nice story. This is such a really nice way when you follow the arrows of learning about a story. And in fact, every nice graphical abstract comes with a series of descriptive bullets that go with the story. And I'm not going to read through these here, but the important thing is that you could essentially draw a line from any of those bulleted words over to any of the lines and pictures on the left, and paint a complete picture of the individual findings on the right and how they led to the big picture on the left. And that's just such a nice way to put things together. So again, you see each of the individual findings, each of the pieces of the mechanism described out on the right, and then put together on the left. And it's definitely okay if you're not following exactly what those findings are. That's really not the point. The point is to show you how this looks and how it works, as you are going to be asked to create a graphical abstract for this particular paper, where we want you to create a summative cartoon or drawing, and then create some summary bullets or highlights that go with it to explain your drawing. And more depthful directions will be found for this within the assignment section here. So this second example, and the last one I'm going to give you, is also from the journal Cell, Host, and Microbe, and you can again see the reference down there, and this was published very recently here. And it involves the bacterial microbiota, and you see a person doubled over in pain, and then a blow up there of their intestine, and you can see this is also a story about regional differences, here focusing on Bangladesh. And within there, you see that these two bacteria are being what's called differentially affected by bacteriophages. And that is quite okay if you have no idea what I just said. I wanted to show you this because of the inherent similarities to what we're doing, but also to show you that sometimes graphical abstracts are accompanied by longer summaries, which almost look like a written abstract. And so again, the goal of your assignment is going to be to put your written abstract that you wrote on a screen or paper, and then make it come to life in a drawing form, and then for you to create summary bullets for the synthesis part, like in the previous example on the last slide. You can hand draw, you can color and hand draw, you could use PowerPoint, you could use Adobe Photoshop, you can let your imagination and creativity run wild. You can draw and take a picture and upload it for the assignment. Whatever you want to do works for us. Or create little cartoons in, again, any electronic software of your choice. This actually concludes the video for the depthful analysis of the article that was assigned to you this week. And I hope this helped sort of get you thinking, get your juices flowing of where to look and how to look, and then to be able to go find some examples of your own as you kind of sleuth through the clues. And we'll see you in class soon. Thanks again for your continued attention and participation.